Good afternoon. Today's topic is bioelectrodes. I'm Dr. Solomon Waniri of the Department of Biomedical Engineering, University of Lagos. I'll be taking the lecture today. So, what are the objectives of this lesson? The first objective is to describe bioelectrodes. The second objective is to distinguish between the various types of bioelectrodes. The third objective is to discuss the properties of bioelectrodes, while the fourth objective is to distinguish between polarizable and non-polarizable bioelectrodes. So what are bioelectrodes? Bioelectrodes are electrodes that are used to sense or measure electrical activity in biological systems. Note biological systems. They are not just ordinary electrodes. They have to be compatible with the human body. So bioelectrodes are electrodes that are used in living systems. For example, in human beings, in rats, in animals, they can also be used in animals. So they are basically used in living systems. A very common bioelectrodes that most of us are aware of is the electrodes that we use in carrying out ECG tests. If you have gone for an ECG test before, to be able to detect if your heart is fully functional, those electrodes that are placed on your chest are bioelectrodes. So the electrodes are able to detect electrical signals from the human body and transmit those signals to the machine that is used to conduct a measurement. So bioelectrodes are used in biomedical instrumentation to measure biopotentials on the human body or on animals, as I've said earlier on. And they can interact with the body fluids. They are capable of interacting with the body fluids. Bioelectrodes are made of conductors. We know that conductors allow electric current to flow through them. So electrodes convert ionic potentials into electronic potentials. So these bioelectrodes are very useful, and as I said, they are conductors. So they are capable of allowing current to pass through them. Now, the currents we are talking about now are currents, electrical currents in humans, which are usually low voltages in the range of microvolts and millivolts. So bioelectrodes are capable of measuring these low voltages and currents and sending the signal to the medical device that is trying to conduct the test. The potentials that are produced at different points can be measured by placing a bioelectrodes at those specific points that we want to measure the signal from. There are basically three major types of bioelectrodes. The first one is microelectrode, the second is body surface electrode, while the third is needle electrodes. What are microelectrodes? Microelectrodes have small tips that penetrate a single cell used to measure voltages. So microelectrodes, based on the name micro, meaning small, so their tips are very small, capable of measuring minute electric current in cells, in a single cell. So they have very tiny tips. They are small enough to permit penetration without damaging the minute cell. So microelectrodes are very small And when they are used to take such measurements, they are able to they are able not to damage the cell. We should note that microelectrodes have very high impedances in the range of mega ohms because of their small sizes. The second type of electrodes is body surface electrode. One of the typical examples of body surface electrodes are the electrodes that we often use to conduct electrocardiography, that is ECG. They are typical examples of body surface electrodes because they are placed on the surface of the human body. They are placed on the surface of the human body. When you go for an ECG test, usually the body surface electrodes are placed around the chest area in specific regions of the body. So they are on the surface of the skin. That is why we call them body surface. We should also note that they have a larger surface area than the microelectrodes and needle electrodes. They have many sizes and types of body surface electrodes. So body surface electrodes, as I've said, can be used for ECG measurements. They can also be used to sense electroencephalography and electromyography. The third type of bioelectrodes is needle electrode. Needle electrodes are generally small and needle-like in size. So their size is like the size of a needle. That is why we call them needle electrode. And usually, one main advantage of needle electrode is that they are capable of reducing artifacts or interferences that affect the quality of measurement. Needle electrodes are capable of reducing artifacts because electrode movement is minimized when we use needle electrodes. And that is why we often use needle electrodes for encephalography when they are placed in the brain to detect electrical signals in the brain. What are artifacts? Artifacts are generally unwanted signals that tend to affect the results of our measurements. Motion artifacts are one of the major types of artifacts. They are noise signals that are generated due to the movement of electrodes. You know, the body, while even at rest, there's a kind of movement in the body occasioned by the metabolic activities that are taking place in the body. There are some kind of movements. So when those electrodes are placed on the body, those movements tend to lead to artifacts that could affect the quality of signals that are measured. Also, the frequency range of artifacts often interfere with signals that we tend to measure. That is why when ECG test is conducted, you notice that they place some jelly on your skin before placing the electrodes. Those jelly that are rubbed on the skin before placing the electrodes on the chest. The purpose of those jelly is to reduce artifacts, the effect of artifacts on the measurement. So when those jellies are placed, they help to establish a low impedance path on the electrolytes. So that is why we place jelly, to reduce artifacts while taking measurements. What are the properties of bioelectrodes? Bioelectrodes have several properties. One of the main properties of bioelectrodes is that they are good electrical conductors. Bioelectrodes are good electrical conductors. We know that conductors allow electric current to flow. So When using a bioelectrode, we know a bioelectrode is supposed to allow electrons to flow through it and send the signals to the measuring instrument. So it's important that we use good conductors. And silver is the best conductor and one of the conductors that is often used. Also, copper is used in the design of bioelectrodes. Then bioelectrodes should be chemically inert. Bioelectrodes should be chemically inert. This means that they should not react with the skin. They are not supposed to cause some kind of interaction with the skin because they are chemically inert. If they start causing irritation on the skin while using them, then you know that there is a problem. They are supposed to be chemically inert. Also, they should be easy to clean because after the test, it's important that we clean the bioelectrodes. They should be easy to clean. They should also have low impedance. Bioelectrodes should have low impedance since they are good conductors of electricity. They should have low impedance. Bioelectrodes also should establish a good contact with the body and not cause motion. Bioelectrodes should establish a good contact with the body and not cause motion. Because when there is motion, then motion artifacts are generated, which will affect the quality of results. Bioelectrodes should not be toxic. They should not destroy the skin. They should not be toxic. Also, potentials generated at the metal electrolyte interface should be very low. The potentials that are generated should not be sufficient to cause electrical shock. So the potential should be very low voltages. Also, bioelectrodes should be mechanically rugged. They should be mechanically rugged, meaning that they should not, for instance, break in the process of using them. Imagine you are using a bioelectrode needle to take measurements on a person, and the bioelectrode breaks and enters the person's skin. You see that it can create problems for that person. So the electrodes that should be considered for use as bioelectrodes should be mechanically rugged and not easily broken. Metals that are often used for electrodes as bioelectrodes include aluminium, copper, silver, platinum, and gold. These are the most common electrodes that are often used for bioelectrodes. Then let's look at electrode-electrolyte interface. When a bioelectrode makes contact with the body, an electrode-electrolyte interface is formed. When you place that bioelectrode on the surface of the skin, there is an interface between the electrode and the electrolytes. The electrolytes in this case are the body fluids inside the body, because the body fluids are electrolytes because they contain some ions. So net current usually flows through the interface from the electrode to the electrolyte. Usually current flows through the interface from the bioelectrode to the electrolyte, and that electrolyte are the body fluids. So the net current comprises electrons flowing in opposite direction to the current in the electrode. We also have cations and anions, that's positively and negatively charged, negatively and positively charged ions. Now let's look at polarizable versus non-polarizable electrodes. What are perfectly polarizable electrodes? Perfectly polarizable electrodes usually behave like a capacitor in the sense that they store charges and do not allow charges to cross the electrode-electrolyte interface, usually when we try to apply current to them. So when we say that an electrode is perfectly polarizable because it cannot allow charges to pass through that interface, because it behaves like a capacitor. Usually a capacitor stores charges and creates an environment in which current cannot easily flow. So that is what happens in a polarizable electrode. While in non-polarizable electrode, current passes freely across the electrode-electrolyte interface. Silver chloride electrode is a good example of perfectly non-polarizable electrodes. So we know that silver is the best conductor of electricity. And when we have silver as a bioelectrode and interface it with a silver chloride solution, then current will freely pass across, and that creates a non-polarizable bioelectrode. So these are some of the references and further readings for this topic.